Hi there, I'm Jakub,
I'm Caitlin.
I'm Emily.
I'm Tessa.
and we're Team 22. Today we'll be discussing our senior design project, modeling nanoparticle drug carriers in a tumor-on-a-chip microfluidic device. In 2018, a little more than 1.7 million Americans were diagnosed with cancer, and one of the main treatments for cancer is chemotherapy. But chemotherapy is far from perfect. Since every tumor is unique, drug treatment must be personalized from patient to patient. And furthermore, the drugs used in chemotherapy are toxic and kill both healthy cells and cancer cells indiscriminately. A tumor on a chip is a microfluidic device that imitates the microenvironment of a tumor attached to a blood vessel. Using the device to measure a drug's diffusion into tumor cells provided by a patient could be the basis for a new personalized approach to cancer treatment. Diffusion of nanoparticle drug carriers, which can protect healthy cells from toxic drugs, can also be tested, helping with managing chemo's nasty side effects. Our goal was to see if we could simulate this all with COMSOL.
The first step in creating a COMSOL simulation is to create the geometries of the tumor-on-a-chip device. We chose N-shape and linear shape designs that included 4 distinct domains: the inlet-outlet vessels, free flow space, hydrogel diffusion chamber, and tumor domains. An important parameter to define in order to add complexity and accuracy to the models is porosity of the hydrogel and tumor domains. Hydrogel porosity is defined as 0.5 and tumor porosity as 0.1. In order to simulate drug diffusion in the tumor-on-a-chip design, laminar flow, transport of diluted species in a porous medium, and particle tracing physics were selected in COMSOL. The model parameters of flow rate and drug concentration, or the number of particles entering, needed to be specified. Inlet flow rate was defined as 0.033 meters per second in the laminar flow physics. This flow rate is used to calculate the velocity field, which determines the particle trajectories. 100 particles were released every 0.2 seconds for 5 seconds, and particle properties such as diameter, sphericity, and density were defined within the particle tracing physics to help accurately inform the calculations of the particle trajectories.
Initially, we studied the diffusion of nutrients and low molecular weight drugs into the microfluidic device and through the porous media domain. A tracer is initially pulsed at the inlet channel. The plot on the left shows the diffusion of the tracer in the free flow region, and the plot on the right shows the diffusion of the tracer into the porous media. As you can see, a characteristic diffusion pattern was achieved for this microfluidic device. The same study was completed on the second geometry, which results in a slightly different characteristic diffusion pattern. With this geometry, we wanted to mimic a tumor that uses a single blood supply for intake and excretion of nutrients. The simulation results of the tracer were validated in the lab. Both geometries were flushed with water, then blue dye was pulsed at the inlet channel. The characteristic diffusion patterns, as seen in the simulations, were verified with these experimental results. In order to simulate the movement of drug carriers, such as nanoparticles, we ran studies using the particle tracer interface in COMSOL. The simulations determine the particle trajectories, which we use to determine accumulation rates on the surface and inside the tumor domain. The accumulation rates at each tumor site in the microfluidic device is shown in the graph to the left. The results of this study can be used to test relative accumulation rates of different tumors seated at each tumor site. The second graph shows the results in comparing the accumulation rates on the surface and inside the tumor sites.
Moving on to the economic summary, the recurring costs are about $7.52 per chip, with $1 of that making up the plastic sheeting for the chip itself, as you can see on the right, and the remainder used to purchase hydrogels and cell culture materials. The capital costs are about $10,200, which buys a COMSOL license, a syringe pump, and a laser cutter. As for the ethical benefits, while traditional drug testing requires animal sacrifice that is not only ethically questionable, but also has an extensive approval process, the tumor-on-a-chip model can reduce reliance on animal testing, as it can be used to confirm in vitro or computer model findings that would normally be confirmed by studies in mice, and requires only institutional review board approval for patient cell use. In summary, the tumor-on-a-chip model allows for easy, cost-effective, and quick assembly thanks to its layer-by-layer design, has the potential to reduce animal sacrifice in drug testing and development, and will ultimately further the development of targeted and more effective cancer treatment, as it allows for the diffusion of nanoparticles to be more easily studied in an environment that is more similar to that of the human body as compared to traditional methods.